Hi everyone, I am Crissa Ann Lilagan and I will be presenting our paper entitled Wide and Shallow versus Narrow and Deep, comparing two transcriptome assemblies of Sila serrata. So this is a paper by Dr. Anish Shrestha, yours truly, and Dr. Maria Carmen Lagman. Alright, so a de novo assembly approach is being widely used in transcriptomic studies of non-model organisms which lack high-quality reference transcriptome. So the accuracy of the assembly is largely influenced by the sequencing depth and the number of replicates. So this brings us to the question, how much data should be generated in an RNA-seq experiment to obtain robust results? So to answer this question, there are several factors to be considered, and these include the number of replicates, sequencing depth, cost, and data storage. So as sequence depth increases, the cost and the data storage increases as well. But if we reduce the sequence depth, we might not be able to recover the less expressed transcripts. So with this, to evaluate the trade-off between replicate count and sequencing depth, this study focused on assessing the difference in the quality of transcriptome assembly between wide and shallow and narrow and deep RNA sequencing data. So the data used in this study are two RNA-seq datasets from gill tissue of Sila serrata, commonly known as mangrove crabs. So each dataset were pre-processed and the clean reads were then assembled through Denovo Assembly approach using Trinity. And this produced a total of approximately 95 million reads spread across the replicates. So for the wide and shallow, we have 6 replicates, which then gave us around 16 million read count per sample. For the narrow and deep, we have 3 replicates, which then gave us 31 million read count per sample. This is twice more than the wide and shallow. So both datasets have 100 base pair read length. Okay, so 3 different quality metrics were used. First off is the contig length statistics. So only the values from the single longest isoform per gene were considered to avoid the skewness of N50 values brought by a large number of isoforms. So the contig N50 tells us that at least 50% of all the assembled bases are found in contigs that are 1077 bases for wide and shallow, while for narrow and deep, a shorter N50 is observed with only 888 bases long. So overall, all length statistics are favorable towards wide and shallow. But what is rather more interesting to show is the EXN50, which is the N50 value of a particular subset based on the level of expression. Now, expression here means the abundance of reads that map back to the assembled transcriptome. So higher expression means higher read support. And here we see a peak of N50 value at 80 to 90% level of expression. And typically what we want is to have long contigs, so that's high N50 value with high level of expression. So in our data, a higher N50 value is seen for the wide and shallow than the narrow and deep. So this means that most of the contigs here are more contiguous at 80 to 90% expression. And lastly, we want to see how many of these contigs align to the protein sequences from SwissProt database. This tells us how much of our data is relevant to downstream applications such as discovery of novel genes and functional analysis. So in general, we obtained a high number of protein entries that align to almost full length, so that's 91 to 100%, to one of the contigs in the assembly. And comparing the two sample design, wide and shallow dataset has higher number of protein entries compared to narrow and deep. So in conclusion, our analysis showed that sequencing more biological replicates with less sequence depth yields better de novo transcriptome assembly quality. We note that this is based on just one pair of dataset and needs to be validated with more data. And lastly, it would also be interesting to see the impact of the two design choices in detecting differentially expressed genes. Alright, so if you have any questions or concerns, you may contact us through email. And I would also like to acknowledge that this study is part of the Mangrove Crab project of DLSU, led by Dr. Maria Carmen Lagman and funded by DOST PICA-ARD. Thank you.